Good afternoon, everybody. Welcome to the workshop on nutritional biomarkers of dietary intake and exposure. Thanks for attending and looking forward to an exciting workshop. I'll tell you a little bit about the workshop and the participants in a second. This was organized as discussion about dietary biomarkers between, as a result of consortium that Oliver Fiehn and Clary Clish and I are members of, and I'll get to that in a second. We're going to hear a wide range of information about dietary biomarkers. We're building on a foundation that has been existing for quite some time. A lot of those existing databases and other resources, however, have not been extremely pertinent to metabolomics. And so part of the task of people interested in metabolomics is to build on the foundation that has already been laid, involving a lot of different dietary components and so forth. In addition, there have been efforts to try to identify metabolite biomarkers of various foods as well as dietary patterns, and some of the work that I wanted to point out was the food ball initiative that Lorraine and others were involved in the mid 2014 to 2017. That set the groundwork for work that has been supported by the NIH, starting with a meeting in 2018 to bring U.S. researchers to get involved in dietary biomarker work. And out of that meeting, proposal RFA was sent out, and a dietary biomarker consortium was awarded a grant from NIH and also from the USDA. That has resulted in the Dietary Biomarker Development Consortium, which is a set of clinical sites involving Harvard, UC Davis, and Fred Hutch, along with three different feeding studies of slightly different designs, and then quite a bit of metabolomics analysis, statistical analysis, and verification and validation, which are important parts of the biomarker development process. So the purpose of this workshop then is to explore some of the issues in dietary biomarker discovery, validation, and talk a little bit about the progress that's been made, some areas of interest and focus for a variety of different researchers, and then, of course, talk about the opportunities and challenges ahead in the area. So the agenda for today's workshop is organized in 4 different topic areas, as you can see here. So I'm going to start with 2 talks about how to find biomarkers, how to develop study designs, what is a biomarker, and so forth. Then we'll talk about how to analyze the data, to acquire and analyze the metabolomics data. We'll talk about putting the data together in accessible databases. And finally, we have a couple talks about outcomes and applications. What we'd like to do during the workshop is, after each of the topics, we'd like you to access this QR code. We have 3 or 4 questions for you to answer. And then at the end, we can discuss your answers to the questions and have a sort of general Q&A. We'll also have time for maybe 1 or 2 questions in between topics. So those talks will stay fresh in your mind. Okay? I think that's it. So, sorry. Let's start with our first speaker, Lorraine Brennan.
Okay, good morning everybody, and I'd like to thank the organisers for inviting me here today just to give a very quick update on some of the work that we're doing in terms of food biomarkers. So I'm going to start with food intake biomarkers and talk a little bit about what I mean by them, and Catalina will pick back up on this in her talk. So first I want to just think about what happens when we eat food. So when we eat food, we actually ingest thousands of small molecules, our metabolites, and these can be further metabolized in our bodies. And this is what we refer to as the exogenous metabolome. And when we talk about dietary biomarkers, it's these metabolites that we're interested in. They can tell us a lot about what people are eating. And of course, when we eat food, it impacts as well our endogenous metabolome. And when we look at the endogenous metabolites, we can start to understand metabolic phenotypes and things like that. But from a dietary biomarker perspective, we're predominantly interested over here on the exogenous metabolome. So we want these biomarkers for a number of different reasons, and primarily the reason is that our self-reported data has a number of well-documented limitations associated with them. And these self-reported data are things such as food diaries, food frequency questionnaires, and 24-hour recalls. And these are what we currently use in nutrition to assess people's dietary intake. We know that people will over-report foods that they perceive to be healthy and under-report foods that they perceive to be unhealthy. So we do need biomarkers. We need them to overcome some of the limitations associated with self-reported data. But I think we need to be also realistic that we're probably not going to get biomarkers for all foods, and we're not going to replace, at least I don't think we're going to completely replace these self-reported tools. And I'm going to talk a little bit about how we can use our biomarkers to improve the accuracy of our self-reported tools. So the approach that we've taken, and we've taken this approach in Foodball to identify new biomarkers of food intake, is to perform controlled intervention studies where we feed volunteers exact quantities of foods, we collect biological samples in a timed response after consumption of the food, and we look to identify biomarkers associated with the consumption of that food. In an effort then to validate some of those biomarkers, we often perform dose-response studies where we're feeding individuals different quantities of the food, and we look to see do the biomarkers respond to the different portions of food. And then if all goes well, then we try to validate our findings in an independent study to make sure that the associations we've seen or the biomarkers hold up in a completely independent study. Underpinning all of this and how to use these biomarkers and how to make sure that they are really food intake biomarkers or dietary biomarkers is validation. And Catalina is going to talk about some of the frameworks that have emerged initially during the Foodball for validating food intake biomarkers and have been further developed since then. So let's say that we go through our controlled interventions, we perform an intervention, we identify a biomarker that's responding to a certain food. It shows a very nice dose response to that food as well. The next question you might ask, well, how are we going to actually use these biomarkers? And we can use these biomarkers in multiple ways, and I'm just going to give you a flavour of some of those ways from a nutrition perspective of how we can use them. And the first one is to measure adherence into dietary intervention. So when we perform randomised controlled trials, we have biomarkers that can really tell us or show that we have adherence to that particular dietary intervention. It would be a huge benefit to the nutrition field. What we really want, or the Holy Grail, is to have objective measures of dietary intake. And again, there are examples of where you can have biomarkers that potentially can give us objective measures. And what I'm going to talk about today, or give you a flavour of today, is how we can use the biomarkers to correct self-reported data. And then finally, a big area is how you can use these biomarkers to study relationships between health and disease parameters. So just to talk about how you could potentially use the dietary biomarkers in combination with self-reported data, I'm going to use an example of work from our own group initially. So we worked on a biomarker of citrus intake, proline betaine, and we showed that you had a very nice dose-response relationship. Once you increase consumption of citrus, you increase the biomarker levels of proline betaine. But what we were interested was to take our biomarker estimated levels of citrus intake, model them with our self-reported data, develop regression calibration equations, and then use those calibration equations to correct the self-reported data when you have no biomarker data. And the equations behind that are shown here. So what you're doing is you're modeling your self-reported data with your biomarker data, and then you can use the estimates then to get a calibrated mean daily citrus intake when you only have self-reported data. And this becomes very important when you move into large cohort studies where you maybe have 100,000 individuals and you have FFQ data for those, but you don't have biomarker data for them, but you would like to improve the accuracy of your self-reported data, your FFQ in this instance. So we did this with around 800 individuals, and you can see that this is the predicted or the calibrated intake, and that's the self-reported intake. So we show that you could do this with biomarker data. So using some simulation studies then, we estimated that you would need about 30% of the population you'd have to have biomarker data for to be able to correct the self-reported data when you only had self-reported data. So this is just one example of how you could potentially use biomarkers of intake, and the concept here being that you're using it to enhance the accuracy of the dietary intake. So there are other people also using this, and this is an example that I've taken from Ross Prentice's group, and Dan is also involved in this work, where they use very similar concepts, where they're using calibrated biomarker intake, in this instance for red meat, to then look at the association between red meat and health outcomes in a larger study. So again, a feeding study, identify biomarkers from the feeding study, and then model the relationship between those biomarkers and the food frequency questionnaire, generate regression calibrations, and then that allows you to get calibrated intake of red meat in individuals where you don't have the biomarker data, where you only have the FFQ data. So now you can start to look at the relationships between food intake and biomarker data in approximately in over 80,000 individuals. And that's what they did, and I won't go into detail in it, but that's just to give you the concept of how you can use it. So there are numerous foods, and you might have seen if you went to breakfast this morning the range of different foods that we have. So it's also, we have to be very cognizant that we're probably not going to get biomarkers for all the potential foods, or indeed be able to identify or perform intervention studies like I described in all potential foods that we're interested in. So we were interested then to see, well, what about if we don't have an intervention study? Could we use really detailed dietary data to try generate some of these calibration equations? And that's what we did, again using citrus as an example. And in this instance, we had a 7-day weighed food diary. So this is really really detailed dietary data. And we modeled it against urinary proline betaine. And here then what we done was we used the biomarker predicted intake, we modeled it with the self-reported data, developed calibration equations from this, and then used it to correct the food frequency questionnaire, which is of lower quality dietary data in a larger sample size. So this was getting over the idea that we might not need to do detailed dietary interventions for every food that we're interested in. And then finally, just to wrap up, I want to say that we don't eat foods signally, we eat foods in combination, so dietary patterns are really, really important. And then when we've been working on this, is to try develop models that will classify individuals into different dietary patterns. Of course, this is difficult, but there are very good examples now that exist where you have multiple biomarker panels that can measure the adherence to certain dietary panels. There's lots of challenges with this type of work, and some of these challenges are going to be addressed as we move through this workshop. And the first one being trying to identify unassigned features. Validation is hugely important, and validation is often not performed, and Catalina will pick up on that in her talk. Dietary patterns, again, I think we need to look at more diverse range of different dietary patterns to try get captured dietary intake in its complexity. And then finally, potentially we need to start thinking that a little bit different about study designs, because we're not going to be able to go through each food individually, and how can we capitalise on some of the data that we have already to try enhance the accuracy of dietary assessment. So I'm quickly going to just say thanks to the team in UCD and thanks to our funders for supporting some of the work that we're doing, and I'll hand over to Catalina and I can take questions after we've finished. So thank you.
Our next speaker is Catalina Cuparencu. We do have time. Do you want to? Why don't we? If anybody has a question now, while Catalina's getting set up, we could take one.
Maybe a question in terms of generalized dietary markers for diverse populations and highly distinct diets. Any comment on that?
I view back to the very first thing where I said we try to get into independent studies if we can, and I think that's really important of seeing if I've done my biomarker work in a certain population group, then I think we need to start now seeing does that hold up in a very diverse population group. So we've started to do some of that work, trying to see if the biomarkers we find in Europe in a very white Caucasian population, if they will hold up when we move to some of our Asian populations. So that's, I think, really important that we start to do that, yeah.
You ready to go?
Yes, I'm ready.
Question?
Thank you. Very nice talk. Always informative for me. So my question is this proline betaine biomarker. I know you saw it like over 1 week consumption, right? But do you think is this marker robust enough to predict habitual consumption?
Yes, because of the second paper I showed where we were doing the 7-day food diary record, and then we looked at its relationship then with the food frequency questionnaire, which was habitual intake, and we had repeated measures of that, and it worked well. So it seems to give us an indication of as good as what we currently have, and gives us an indication of habitual intake, yeah.
Okay.
Can you hear me well? Good. I would like to start by thanking the organizers for having me here. And I've been working with biomarkers of food intake ever since my master's, that's 12 years ago. And I was fortunate enough for my PhD to work in the football project, and devoted those years to truly work with biomarker validation and the application of biomarkers of food intake in nutrition studies. So what makes a good biomarker? When we're talking about biomarkers as dietary assessment tools, we're really interested to reflect the exposure to the foods and dietary patterns we consume, and not the effect of these dietary interventions, and that is really, really important. So ideally, when these biomarkers as dietary assessment tools are non-nutrients, and there are some metabolites that our body is not really interested in. So if we can just excrete them the way they come in, would be perfect. So within the football consortium, we developed a framework for validating these biomarkers that covers a range of biological aspects and analytical as well. And I will now take you through all these different criteria. So the first one is plausibility. And when we talk plausibility, we want the biomarker to be chemically related to the food of interest, but also specific. And usually when we have all these untargeted metabolomic discovery studies, we have many suggested candidate biomarkers, but these should undergo a critical biological interpretation. This is in order to prune the final list of candidate biomarkers, but also to have a good understanding of the different sources of variation. And the current challenge with this criteria is that we have many candidates that relate to foods, but most of them are not specific. The second one is dose response, and Lorraine touched very nicely upon this concept. And here we want to see that the BFI increases along with increased intakes of the foods. And in those response randomized controlled trials, we can develop calibration equations that we can then use to assess the quantities of food intakes in observational studies. The third one is time response, and this is really, really a crucial validation criteria. This indicates the half-life of a metabolite, as well as the elimination kinetics of this metabolite. It also should indicate whether, when we repeat consuming the same foods, would then this metabolite be saturated. So these kinetic profiles are really essential to determine the window of sampling. So when we have would this biomarker be present when we sample? And when we have a random sample, what do we expect to actually see in that sample? Would we see a biomarker of habitual diet or one of recent? So in the time response studies that we do in discovery, we usually I show here 2 examples of meat biomarkers, and you can see that for carnosine, for example, this biomarker 12 hours is already back to baseline levels, whereas for 3-methylhistidine, it's already still increased at 24 hours. That means when we have a random sample and we measure carnosine, we're quite certain that the meat was consumed in the day when the biological sample was collected. Whereas for 3-methylhistidine, it can indicate a bit of a longer intake. The next criteria is robustness, and with this criteria, we're interested to see that the biomarker works as part of a varied diet. And that's mainly because when we develop biomarkers or discover biomarkers in highly controlled studies, we control the diet, and we don't really know the other sources of variation. So we would like to fulfill this criteria by confirming biomarkers, at least replicating them in studies with similar designs, but ideally validating them in studies of contrasting design. So from a randomized controlled trial to observational studies. And a lot of the non-specific or endogenous metabolites will also fail this criteria as a result. Last but not least, reliability, and here we want to compare biomarkers with other dietary instruments, ideally with other biomarkers. We want to be there, but so far we can, at the minimum, have associations between biomarker levels in blood and urine and intakes from dietary registration, which is what in nutrition we call the relative validity. And ideally, we would actually use these biomarkers to predict intake in independent studies. An accurate reliability, however, relies on the comparator. So when we have an uncertain biomarker that we're just developing, comparing to an uncertain dietary registration, we don't really know who's failing, so then we have to repeat this many different times. And the excretion kinetics, as I mentioned earlier, it's really important. So this reliability should be tested in biological samples that reflect the time window of sampling of the said metabolite. So by applying these concepts, we can actually see that from 14 candidate biomarkers that are already plausible and have a nice excretion kinetics, only 2 are predictive of intake in an observational free-living population. And among those, 6 are validated. And this is for meat. Analytical aspects of the validation imply the stability, so the biomarker must allow repeated measurements with no degradation. In observational studies, we have a bunch of big cohorts that were using samples from over and over again, so we need to have biomarkers that can withstand the different thaw-freeze cycles. Regarding the analytical performance, usually for biomarkers detected in untargeted metabolomics, this is already fine because when we then have targeted methods, usually we can measure the abundance a lot better. But in order to develop targeted methods, we need internal standards and limits of the quantification, and I think the next speaker is going to talk a bit about how we can improvise there. And not least the reproducibility between labs. So it's important that it's not only one lab that can find a metabolite, but that we can standardize methods across laboratories and push this forward. Recently, we added another criteria that we overlooked, or it was not really flagged in the validation framework developed in football, and that is biological variation or variability. So far we actually don't, when we conduct the discovery studies, we can see usually some sort of a variation in biomarkers level within the groups, but we are not really certain, or there's no evidence to say that even the same person eating the same amount of food will actually excrete the same level of the metabolite. So for these biomarkers to actually work, we need a low intra-individual and inter-individual variation in biomarker response to the same amount of food consumed. And this has so far, it's largely unexplored, but it's beginning to come forward by applying ICCs used in samples collecting longitudinally. Usually we have around 2 such of these samples that were based ICC scores on, but those are still influenced by half-life, frequency of intake, and the overall dietary habits in the population studies. So experimental studies actually tackling this are really, really needed. So the big question is, should all biomarkers fulfill all these criteria? It's a bit overwhelming, and this is how we felt after football as well. And that would be ideal, but you know, the biomarkers are, this validation is not really meant as a score, so it's not how many of those you have, then the better the biomarker. These criteria are meant to actually characterize the biomarkers, and then we can know how to develop new approaches and how we can actually use this forward. So here I show an example of a qualitative prediction of red meat when we actually have no specific red meat biomarkers. And we use a ratio of anserin, which is a marker of chicken, and carnosine, which is a marker of overall meat intake, to discriminate between red and white meat in a free-living population. And we use samples from a postprandial studies on the left-hand side to predict samples in observation or in free-living individuals with quite high accuracies of area under the curve of over 0.9. So a sufficient characterization of BFIs and testing them in different studies to allow new ways of combining biomarkers is essential to accelerate their implementation in nutrition research. And if you're interested in hearing more, I'm also having a talk during the Nutritional Metabolomics session on Tuesday, where I'm going to present some of the concepts that we are put out in a nice review that is currently in press in Nature Metabolism, where we tackle kinetics, frequency of food intakes, and not least important, multi-sampling to push the field forward. And with this, I would like to acknowledge my group at the University of Copenhagen, led by Professor Lars Draxet, as well as collaborators in football and beyond.
Okay, we have time for a question or two. Thank you. Very nice talking. Great.
Thank you for such excellent summary. My question is regarding really the reliability criterion, and you touched a little bit on that. I think Phil would remember we did a critical review of current biomarkers in the literature since inception, and at the time we really struggled to check whether biomarkers satisfied this criterion or not. So practically, as you say, there aren't enough gold standard methods to compare that, so it seems like this criterion is really not different than just seeing if the biomarker is specific or not, doing correlation with intake. So do you have any comments or insights on that specifically?
I mean, it's a good point, because in a way there are all these different criteria that are slightly intertwined. So plausibility and robustness, you already see robustness a part of observational study, then you would usually then already confront them with dietary information that you have from the observational study. So in theory, you could say reliability is ticked, but it's ticked just to some extent. So I think as a first step, that's already good. But where we want to go forward with is to actually predict in independent studies. So to use many more of this observational study, not only one to test the reliability and robustness, but more. And then in our experience, you will see that the list of biomarkers decreases. But in a review that we also just wrote, then at least, I will say it's still reliable and requires forward validation.
Thank you.
Very nice talk.
Thank you.
So my question is about your 9th criteria that you talked about.
Yeah.
Inter-individual.
Yeah.
So do you have any potential markers in mind to cite as an example for that?
Well, I think, the good old standing proline betaine that works in everything could probably be, because we have, it's already quantitative at a very large number of individuals. But, and in those response studies, you are actually having the same participant eat the food over and over again. So I would say that those responses would touch upon the variability as well. But we don't have many. It's largely unexplored, and not even for the, the better of the biomarkers, we don't really know.
So my inclination is based on the the description what you provided. It could be something like a microbial metabolite that metabolizes early in some individuals versus, like late. So it shows up in different times. So that might fit into some.
Indeed, but then it's also that is dependent on the individual microbiome. So different people will have different microbiomes. So we don't really know if that biomarker would end up being robust in the same way. So I think the going forward from here is to have experimental studies with the same amounts of foods within the same individuals to test some of these biomarkers that are most known and see, what we see to get real evidence for it.
Okay, I'd actually like to move on, and then let's write down your question and save it for the discussion, because we've got quite a bit of an agenda. Can you put this on the screen now? So we're going to go to the survey questions, and apparently some of you have already answered those, but you may want to rethink them a little bit. So if you haven't copied or used the QR code, please do so now, and then answer the, was it, Oliver, 3 or 4 questions for this session?
Per section is 3. So the idea is to do section by section, but don't submit. You can only submit once. Once you have submitted, you can't go back. So the idea is you submit at the end.
Okay. Yeah. And our next speaker is going to be Oliver, and so he'll get set up while you answer the question.
All right. So let's. So that was the idea of it all, and now how do we do it? How do we even measure things? Which is kind of cool in the metabolomics intro. Open. Okay. Metabolomics conference. People think they all know, right? Maybe we do. And how do I bring it online here? Okay. F5, how do I? Oh, shit. Sorry. This is a PC. So how do I bring it to full screen? Don't know. That's a PC.
Oh, yeah. It's normally over here somewhere.
Slideshow? All right. That sounds about right. From beginning. There you go. Thanks. Okay. So how do we analyze and annotate dietary biomarkers in the first phase, the discovery phase, not the validation, not the quantification. How do we do this even? And the idea is to do it right and to share. Okay, so that's the take-home message. In terms of how to measure things, it starts, of course, with a good study design. We talked about it already a little bit. No good study design, no good biomarkers, that's for sure. And then at some point you get samples. And what you do with the samples. So, as we learned often, it is important that the biomarkers are reflecting the foods that we are looking at and should be different to the components, the endogenous responses to it. Now, obviously, the endogenous responses are magnifolds higher, specifically for lipids. So we have, a good choice would be to do lipid removal first. And that's the same, by the way, for exposome compounds. All right, now there are some papers on how to do it. I read all those papers, and I didn't find them very convincing. So there's still some room for method development to do better methods than those, right? But these are the ones that are online right now. So on the one hand, you would have hybrid SPE cartridges. That's a paper on the exposome, but it would also apply for food biomarkers. Then we have the ostroplates from Waters. Again, 2 papers that I listed there. The Captiva columns from Agilent, and we will hear more about those in the PTFI use cases. And then the free cartridges from Phenomics, which you still have to pay for, although they are called free. So this is all fine, and they kind of work, but they also, of course, take away things. These are not cartridges that only take the phospholipids and the triacylglycerides. A side comment from myself, if you just want to get rid of the triacylglycerides, that's easy. 50% acetonitrile will do the trick when you do a resuspension. Triacylglycerides will not like it. But the phospholipids would. Okay, so there's that. And then the next question, do we need special extraction, chromatography, or MSMS methods for food biomarkers? Well, first of all, different HPLC methods or GC methods, or even NMR methods, would look at different types of compound classes. We all know that in this room, right? So usually, for that reason, you could say that one method is not good enough. I think in metabolomics we agree on that, don't we? We will see if there's a consensus. That's maybe what the survey is about, right? And secondly, if we talk about compounds, I want to be able to share data and could be even unknowns, right? Because what do we really know about foods? We will hear that from Steve Watkins, what we do know about foods. But if we want to make sure we find the same compounds, maybe even the same unknowns that other people have reported, we have to use standardization. And in order to standardize methods, the most important thing to standardize is the retention time. And to be able to talk about retention time in GC or in LC is to use the same set of internal standards. If you use different sets of internal standards, it's going to be very hard to compare. But once you do it, it's easy. In GCMS, it's done for 70 years now. Dr. Kovacs has done it. And in GCMS, everybody's like, yeah, it's called the Kovacs Index. What are you talking about? It's easy. In LC, there are a couple of papers. One is there using these alkyl sulfonates. Unfortunately, these are not commercially available. It's a really nice paper, and there are other ideas to do these internal standards that cover both helic and reversed phase really nicely, but they are not really commercially available. Great idea, though. Maybe at some point they will be. The idea then is, and we published that, is once you have a very clearly defined set of markers, standardized markers, then you can use machine learning to exclude compounds in the compound ID sphere that are beyond that retention time. So that's why we do it. That's another reason why to do it, right? Okay. So is it now difficult to identify food biomarkers, beyond those that are in the libraries already, where we have the standards, you can buy them, you can put them in, a proline betaine, you just buy and you're sure. But what about the others? So in principle, what we all do is we have an MSMS spectrum, you look at the peak here, and it's fine, and then you assign an identity, and often this identity is, especially in NIST libraries, is a fragment. And so you're not sure, so that's an in-source fragment. There is a real thing and it matches something in the library, doesn't mean it's identified. So it's something like that or something similar. So the next thing is, as we talk about plasma, also urine, often these dietary biomarkers will be very low abundant. And so the lower abundant, the more noise ions you will have on the MSMS level. So you have to denoise it. It's unpublished work, but from that paper that we haven't published yet, that's already, we will have a method out soon to really massively improve these low abundant MSMS spectra to take the noise out. And we have also in that paper that I don't talk about really, but it's important to do it, and there will be methods coming out soon, okay? And the third one is quite a number of people are then using that information of MSMS and propagating likely annotations. So for example, neutral loss searches and in silico predictions, and we've done that on a number of compound classes, but also other people, glycosides, glucuronides, sulfonates, sulfates, phosphates, and so on. Modifications of classic metabolites that we see in the food, but that are then modified by either microbes or by the liver enzymes. They are not in libraries, so we have to use these in silico tools. And here you see from a paper that we published last year, on the left-hand side, how many more compounds there are than before. So then, what is a good match? Well, depends on who you ask. If you ask Peter Mikler, he'd say, well, you gotta believe the retention time, because the MSMS will lie into your face. Okay, so it is important, and that's why we need these identifications. I will give a talk about that in this session, not in this session, in the conference. So there are universal identifiers and standardized repositories. For example, mass.wiki, massbank.us, massbank.jp, and .eu, as well as GNPS. And next, we really have to quantitate compounds. So how do we do this when you cannot buy a compound? How do we do this? Well, the first thing is always use internal standards. And with these internal standards, you can, if it's a known compound, you can use a dilution curve. So it's called isotope dilution, and that's the gold standard. People have done single-point quantifications. You can do that too. But more importantly, you can also do things like sample dilution, so to be sure that you have some idea where it is. And we recently have shown at ASMS last week, but it's also got unpublished, coming up soon. When you have many, many, many internal standards, you can group them into chemical classes. You can have them already in an internal calibration curve and use those to estimate concentrations. So the easiest thing is, of course, for lipids. Okay, right? So that's what this shows here, the lipids, lipid quantification, possible. For non-lipids, harder. But there's a number of ideas how to do it, and that's the next frontier for metabolomics to solve. Quantify without standards, or using all sorts of both internal standards and machine learning. All right, so conclusions. Phospholipids, neutrolipids need to be removed. Ion suppression is real. Also in-source fragmentation is real. So retention times are key. You have to standardize it. It's done in GCMS for a long time. We need to move on, and we need to do that also in metabolomics to be able to share. High quality MSMS is critical. I'll talk more about flash entropy and mass.wiki later, but the predictions of modifications of metabolites are possible today, and also MSMS denoising, and then public sharing with mass.wiki, GNPS, massbank, and so on. All right, so now please, you can go already to the survey and go for the next set of questions while I keep on talking. Just for the heck of it, while you do it, we also tell people how to do these things. There's an international summer course that we invite you to. There's another barcode if you happen to like it. Okay, that's from my side.
Thank you very much. We have time for a question or two as Stephen gets ready to talk. Question, anybody?
Any question or comment? Right away? Then it was clear. Thank you. Okay. Oh, you have your own?
Yeah, I think I'll figure it out. All right, well, sorry in advance that I'm not David Wishart. He apologizes for not being able to make it. Let me just find his talk. Clary, I don't know where. Doesn't show up? This may be it. Yep, got it. I just had Clary and Dan Get a little exercise. Anyway, so I'm going to give David's talk for him, but I know very little about David's work, certainly not enough to actually give a talk for him. So he sent some text, and I'm going to read it to you. He'd like to apologize for not being here. He's been delayed with a series of, yeah, I'll get it. Yeah, thanks. With a series of flight delays and cancellations, and I can tell he's frustrated because this next sentence is, I should be there tomorrow if my flight doesn't get canceled again. So anyway, he's asked me to present this on his behalf, and there's a talk on data on databases on food components and biomarkers. So for most of the last 50 years, various national agencies have been measuring and reporting on the composition of local foods and dishes. These food component or food composition databases cover thousands of foods, but typically only provide data on about 30 to 40 dietary nutrients and components. The focus of these is really on macro and micronutrients. They don't provide any detail on compound structures or organoleptic features, health effects, or chemical structures. From a food component or metabolomic perspective, these are what we call shallow coverage databases, and they cover only a tiny slice of the food metabolome. So to address these problems of shallow coverage, several scientists have independently compiled data on the nutrient and non-nutrient chemicals in foods. This information is found in Dr. Duke's phytochemical database, the Dictionary of Food Compounds, and the EAFUS database. These provide data on more than 30,000 compounds. However, the databases are pretty old and don't provide much data on compound structures, descriptions, health effects, or concentrations. So while these databases provide deep coverage, they're not very useful for metabolomics research. So given the shortcomings of present food component databases, there's a real need to rethink what should be in these databases. Furthermore, given the growing number of studies using metabolomics to look at food components and intake biomarkers, there's a need to create databases that can house this metabolomic data and which can be used by metabolomics researchers. This means making the databases smarter, more modern, more aligned with the needs of metabolomics. I'll talk about 6 databases which are addressing these shortcomings. That's HMDB, PhytoHub, Food Atlas, FoodDB, Exome Explorer, and MarkerDB. While most of you may not think that HMDB or think of it as a food or food intake database, it actually houses an enormous amount of data on food and food intake compounds. Roughly 70,000 of these are in HMDB. It also contains experimental and predicted spectra for all of these compounds and supports all of the searching functions that have made HMDB so popular with metabolomics researchers. The plan for the next year is to begin offering support for user deposition of reference compound data. 2 lesser-known databases designed for the metabolomics of food are PhytoHub and Food Atlas. PhytoHub was developed by Claudine Manach in France, while Food Atlas was created by Ilias Tagapoulos at UC Davis. PhytoHub has data on phytochemicals in foods and their metabolites, and is designed very much like the HMDB. Food Atlas, on the other hand, makes use of large language models to extract data from food databases and the scientific literature to create data about food components and their health and disease effects. Much of the data in Food Atlas was obtained from FoodDB. FoodDB has been around since 2012 and is the equivalent of HMDB, but much more focused on food compounds. It currently has about 70,000 compounds from more than 800 foundational foods, and technically it's the largest, most comprehensive food compound database in the world. It's been specifically designed to serve the metabolomics community with information on spectra, organoleptic data, health data, disease data, food association data, which are all entered into this database. A major update for the database is expected to be completed late this year. We've mostly focused on food component databases, but an equally important aspect of food metabolomics are food intake biomarkers. This information is being compiled not only in the HMDB, but also in another database called Exome Explorer, which was developed by Augustin Scalbert as part of the Food Ball project. And a new update is expected here in 2025. Last but not least is another database that Dr. Wishart's team has developed called MarkerDB. This houses extensive data on tens of thousands of biomarkers. It includes plenty of chemical exposure biomarkers and, importantly, 135 dietary intake biomarkers. It's very well annotated and contains a plethora of data covering every aspect of biomarker knowledge. A new version of MarkerDB will be released this year. These screenshots are actually from the new version. So to conclude, David believes there are now a number of new and newly designed databases that are here or on their way. These will support the deposition, identification, exploration, and discovery of food components and food intake biomarkers, and they also make use of plenty of new AI and machine learning techniques, and will provide depth and breadth that the new generation of food researchers really need. Thanks. I don't think I've ever read somebody else's talk before, so that was fun. Should we, given that I'm not sure I'm the best person to answer questions for David, should we just jump ahead? Okay. All right, now I get to give my own talk. Okay, this is about the Periodic Table of Food Initiative, which is a really exciting global initiative that was started by the Rockefeller Foundation and the USDA and is led by the American Heart Association that I've been involved with now for about 4 years. So the idea and sort of the concept behind the PTFI is that it wants to build the world's deepest and best and most comprehensive database of composition of foods. It goes beyond metabolomics. We have lipidomics, we have glycomics, other aspects of the project. But for metabolomics, the key things were really these concepts. One, we wanted to create this central deep database, but we wanted to do it in a way that would allow labs around the world to participate in the generation of data. And I think we all know that sequence-oriented analytics, gene sequencing, transcriptomics, proteomics, these are much more advanced in terms of their standardization and their democratization than metabolomics and lipidomics. We still live in a world where every lab sort of has their own method, and it's very difficult to integrate results from different labs. But a central tenet of this project was to develop analytical platforms that anyone, anywhere could use to participate and contribute to the food database. And that really meant that we had to think a lot about driving data quality and standards that allowed those data to be comparable across labs and geographies and time. The network is already global. As I mentioned, this was started by the Rockefeller Foundation and the USDA, but with the American Heart Association and the BIOT-CIOT Biodiversity Alliance leading the way, we've onboarded labs around the world, I think on every continent except Antarctica. And it's a growing network of labs which are using these methods to contribute to the food database. So this is kind of the starting place. When I first joined, the PTFI had a set of 12 food samples that we sent to, 20, 25 labs around the world in blinded duplicate. And these are 3 very good metabolomics labs that were looking at these 12 samples in duplicate. And then we asked everyone to annotate the data as best as you can, tell us what are in these samples, and send us back the final results. And what we got back was results that made it very clear that these labs could reproduce their own data. They were very good at reproducing the blinded duplicates sample to sample. But when we looked at the annotations, there were almost no overlaps of the identified compounds between the labs. Right? So we were getting less than 1% overlap between all 3 labs, less than 2% overlap of the annotation in any 2 labs that we looked at. So clearly there was a standardization problem. And as Oliver has mentioned in the last talk and others have mentioned, a lot of this had to do with the variability in retention time and the incompleteness of the quality spectra that are out there for annotating foods, and sort of the way those things interacted made annotation sort of chaotic. So this is where we focused on development. There's really 4 technologies. One was we didn't develop new protocols for analysis. We adopted existing protocols that had proven themselves robust. We have a reverse phase metabolomics method from Nicole Reisdorf's group at Colorado. We have a Hillock method from Oliver's group at UC Davis that we didn't modify too much. We basically adopted those methods, but we did invest in developing and manufacturing standardizing reagents. And for each method, this is about 30 internal standard or retention time components that we've manufactured and we distribute to every lab in the network to include in the extraction of every food sample. And that allows us to accept raw data from our labs in a centralized data pipeline and to time align everyone's data before we begin the annotation process. Another key aspect of the annotation was we spent a lot of time rolling up features into chemical formula so that everyone's output in the process is a formula at a retention index for a food sample measured. And because of the scale of the initiative, because we have labs all over the world and the investment in it, we sent every sample and every authentic compound we were building a library around to at least 2 labs in the network. And when we saw the same chemical formula at the same retention index in the same sample, that became a PTFI library entry. It means we expect this is expected in this kind of sample, and it became something that we look for going forward. What's interesting is when we look at those library entries, this is a little bit like a sequencing depth plot where we grouped the foods we measured by botanical family or food family and asked how many compounds are we seeing in consensus across labs when we run these samples as a function of how many samples we're running in each food group. And you can see with, I don't know, laser pointer's not there, it's pretty weak, but animals and dairy, where as we measure more foods, we're sort of flattening out around 4,000 compounds that we're seeing reliably in these foods, whereas things like legumes and potatoes, tomatoes, et cetera, have more than double the amount. And we're still discovering new sort of chemical space as we measure more samples. So it's a pretty rich, very rich environment. And where we are now is that we have all these locations, and now we're beginning the process of figuring out what all of these compounds are. And for that, we have, as I mentioned, on every sample run, we've got a chemical formula, a retention index, and then we see its distribution in food, so we know sort of the genetics of its origin. But we've also run, because we time align all the data, we can run the same sample, collect detailed collisional cross-section information and MS information on every food sample. And that all gets sewn up into a time-aligned file of all of this information around each food. And that allows us to begin identifying what these components are and identifying how many of them are known to science and how many of them are unknown. All of this is being reported in a public database. We've got a sort of 2 sides of a database. One is the more traditional food components that you might see in Food Data Central, lipids and fatty acids and amino acids and sugars and fiber types. And then we've got a discovery side as well, which is reporting all of these new novel metabolomic chemical features that we're finding in foods that you can go and browse them and see the distribution across foods or compare one food to another. And that's where we are. So thank you very much. Oh, and Oliver would like me to say, complete the survey. I'm not going to push pull you like Oliver, though, tell you how to fill it out.
All right, do we have time for a couple questions?
Good.
You were looking at 1,000 foods, or what's the total number of foods that you're...
So we have done the first 500 what we call reference foods, which were selected. Oops. We've done the first 500 what we call reference foods, which were selected by a network of botanists around the world who want to contribute different sort of genetics to it. But we've run, I don't know, another 500, another 1,000 in what we call demonstration projects. Somebody who's interested in quinoa and regenerative agriculture or carrots and this. Or we've done, because the American Heart Association is involved with the Food as Medicine program, we've also done a fair bit of serum profiling as well. A few thousand samples. But 500 are what you'll see if you go to the database at the moment.
Thanks for a nice talk. I actually have a range of questions, but I will stick to one. What is the timeline of this? And what is the timeline of this? And in the discovery studies, do you also, are you going targeted or untargeted in the compounds within the foods? So do you measure what you already know or also new things?
So this is just one, well, I just presented one work stream within the PTFI. We have both targeted and untargeted work streams in the program. So I presented what the metabolomics untargeted, it's sort of untargeted, although once we build a library entry for a known compound, it's sort of a semi-targeted approach. We launched all of these tools and the databases at an event in April in New York. So if you want to participate and onboard these methods in a lab and get access to the reagents, you can just contact the PTFI and do so. So that's the timeline where we had a launch in April, and now we're standing up all of these national labs that I showed in the slide that are going to begin generating data on new samples for the PTFI. And then others who want to raise their hand to participate, there's a process on the PTFI website for becoming a member lab.
Did I hear correctly that you're taking the advantage of other omic efforts also in addition to metabolomics?
Yeah, we have a lipidomics program, which is a sort of a massively targeted program. We're measuring about 3,000 complex lipid species quantitatively. We have a glycomics program, which is isolating fiber and then looking at the sugars and the glycosidic linkages. We have an ionomics mineral profiling program, which I think like 30 minerals. And then we have a bunch of targeted assays like fatty acids and amino acids and that are more traditional nutritional components. So there's a bunch of components to this.
Okay, so because I just wondered, these days even the DNA-based approach is also gaining momentum, but in, like a biological matrices. But so I was wondering whether taking the advantage of such an approach, to your periodic table thing, might reduce the dimensionality of the data what you are looking at.
Yeah, we do have some proteomic. We do a fair bit of proteomic profiling. The genetics ends up being tricky. I'm not the person to speak to this, but the international treaties around moving food materials from country to country and doing analytics and making public databases is very tricky. And I think I let the Rockefeller lawyers work through all of that. But I think the genetics was a particularly tricky part of trying to integrate data into this. But I know that some of the example projects that I mentioned are doing deep gene profiling as well in combination with some of the phenotypic stuff.
And for all you know, you might have the data already in some databases. All you have to do is maybe plug it in.
Yeah, one thing I neglected to mention is that there's a concerted effort to collect a lot of metadata here as part of the database. An apple's not an apple. Where did it come from? How was it grown? Can we link to weather databases? So there certainly is there's a large component of that as well to mine through.
Okay, thank you very much, Stephen. Our next speaker is Clary. Clish.
All right, well, thank you. So we'll begin the next topic, which is on outcomes and applications. And yeah, this is kind of weak. So I'm going to be talking about metabolic signatures of dietary patterns and associations with disease risk. So this is an open question, I guess. Oops, sorry. Can we identify metabolic signatures of dietary patterns, and do they help in disease assessment? Well, we know that some diets are more healthy than others, but is it possible to assess adherence to these diets, and can that be associated with outcomes? I'm going to use an example of some work we did with the Mediterranean diet. And the Mediterranean diet is characterized by high consumption of fruits and vegetables, seafoods, nuts, legumes, whole grains, and olive oil, with moderate intake of wine, mainly with meals, and then lower intake of things like red meat, processed meat, sweets, and sugary desserts and beverages. So going back over a decade, we've been collaborating with a team in Spain that ran a large diet intervention study called the Prevención con Dieta Mediterránea, and I apologize, I don't really pronounce Spanish well, but it's easier for me to say the PREDIMED study. And in this, the goal of this project was to determine whether or not adherence to a Mediterranean diet eating pattern would prevent cardiovascular disease in a high-risk population. So in order to address this, about a little over 7,000 participants were enrolled between 2003 and 2009 at about 18 different sites in Spain and allocated to one of three diets. Two were Mediterranean diets, one supplemented with additional extra virgin olive oil. Both diets had virgin olive oil in them, but one had a little more. Or a Mediterranean diet supplemented with nuts. And the participants were given both the olive oil and the nuts. And then the control diet was the American Heart Association recommended low-fat diet at the time. So as part of this, this trial, the participants were screened at baseline, 1 year of follow-up, and then quarterly using a 14-item dietary questionnaire referred to as the Mediterranean Diet Adherence Screener, or MEDAS, to assess adherence to the study. So among these 14 questions were questions about, how much of these components do you eat? So the score was measured on a, 0 to 14 scale, and a score of less than 10 indicated low adherence to the Mediterranean diet. So overall, the trial or the study was successful. So in less than 5 years, the investigators who were led by Raymond Neustrix and Miguel Martinez-Gonzalez demonstrated that people who were on the Mediterranean diet eating patterns had lower incidence of cardiovascular disease. So we've done, our lab was fortunate to work with these investigators to find metabolic indicators that predicted risk or incidence of cardiovascular disease, type 2 diabetes, and then subtypes of those indications over the years. But for the purposes of developing a signature, we were able to use data from roughly, 1,800 baseline samples and 1,500 year one plasma samples that we had analyzed already using Metabolomics. And among these samples, there were 370 incident cases of cardiovascular disease. And the score, elastic net regression analyses were used to find metabolites that were correlated with the MEDAS scores. And then for replication, we used data that we had also acquired across actually 10 different independent projects in which we had samples from participants in the Nurses' Health Study, both 1 and 2, in the health professionals follow-up studies. So across these 10 projects, we had data on approximately 7,000 participants, and among those, there were 351 incident cases of cardiovascular disease. And again, the same type of analysis was applied using a kind of a regression analysis to find metabolites that were associated with the score. And the score here was derived from data from food frequency questionnaires that were given to participants in those studies. And I believe the frequency of the questionnaires was every 4 years, because it's longitudinal, both longitudinal cohorts. So what was found was that we could measure a pattern of 67 metabolites that were associated with the MEDAS scores, or correlated with the MEDAS scores. Most of the components were lipids, but there were some polar metabolites from our amino acid profiling method and some others. And shown here are I keep hitting the wrong button here, are the correlations between the metabolite signatures and META scores for different groups of samples. So these are the PREDIMED baseline samples, the PREDIMED year 1 samples, and the Nurses' Health Study and Health Professionals follow-up studies showing a kind of a positive correlation between the signature of 67 metabolites and the META scores. So, and in addition, we could look at just these metabolites themselves. Again, remember that the people who were randomized to the Mediterranean diet in the original study had lower incidence of cardiovascular disease. So the question is, could we then just look at this signature, 67 metabolite signature, and see whether or not there was an association with incident cardiovascular disease itself? And that's what's shown here in both cases, people who had higher Mediterranean, in which this pattern was more consistent with the META score, those people had lower incidence of cardiovascular disease. So in this particular case, we could use the metabolite score as a surrogate for the records on compliance to the Mediterranean diet eating pattern. But one thing that's important to point out is that these data were, the metabolomics data that were used here were from a limited number of platforms and data we acquired a while back. So of these 67 metabolites that were in the signature, they were largely composed of generally measured plasma metabolites. So many lipids that we measure in everybody, and many polar metabolites that we measure in everybody, and essentially no known food-specific biomarkers. So these were just basically, endogenous, or essentially endogenous, or very common metabolites, if nothing else. And again, just one thing to point out is that some of these metabolites, although let's see if I can get it with this, no. Some metabolites, of course, were lower, showed a negative association with the diet, and others showed positive associations with the diet. It's a little different than a biomarker per se. So going back to this question, can we identify biomarkers of dietary patterns and do they help in disease assessment? I'd say that we can identify metabolic signatures of diet and that they are associated with outcomes. But a limitation here is that the discovery and replication groups of samples that we use included thousands of samples. So it's a fairly laborious way of getting to such a signature. The questions are, how could we potentially improve the approach used here? And, obviously, it would be good if we were able to include more annotated metabolites. So we've since annotated more metabolites, even in those methods that we ran at the time. And to Oliver's earlier point, we should really, it'd be ideal to include more profiling methods. Here, these metabolites largely came from a couple of methods. We did not include any data from robustly measured unknowns. And as Steve pointed out, there's quite a bit of, we only know the identities of relatively few compounds that we can measure. And then lastly, ideally it would have been nice if we had pre-identified food-specific biomarkers from the Mediterranean diet components and included those in the analyses. And I think if we had those sorts of data, I think our score might have been perhaps a little bit more sensitive and probably, hopefully more specific as well. But I'll just stop there and I'm happy to answer any questions or hold them until after Dan's talk.
Questions? Yes.
Sure. Thank you. Great talk. I'm curious of the study population in the two trials, whether they are similar and whether they are at risk of the cardiovascular disease. And I'm asking that in relation to, did you try to do the same associations but baseline samples, so not after 1 year of the intervention of the Mediterranean diet? What are your thoughts on that? Because the assumption of these people actually consuming for 20 years on the same diet with the same profile, also in the nurses' health study, is quite a big assumption. So I'm wondering, how do you think about that?
Yeah, so good question. So in terms of the PrediMed cohort was, and I didn't put up the clinical characteristics, but these were people that were deemed as being at risk for cardiovascular disease, whereas the health professionals follow-up study and the nurses' health study enrolled over 100,000 health professionals of various states of health. So while there may be overlap between the two populations, the enrollments were a little different. So, and then second, your second question was regarding the timing of the samples. Is that, so yeah, so I believe the food frequency questionnaire data was used from the time around which the samples were acquired. So as opposed to longitudinal data, because we were looking at, again, we needed, the goal here was to assess the metabolite profiles measured in the samples with dietary information that was known at the time those samples were taken. But in order to look at cardiovascular disease incidence, obviously you have to have follow-up. And I think maybe I don't know if I'm really addressing what you're asking. So was your question like, how do we know that people remain compliant with the diet, say nurses' health study, all the way through?
I think my question is related to, since what you measure is lipid-related and endogenous metabolites, my question is, isn't this study population more prone to have this signature that is then linked to CVD risk instead of actually the real diet?
Yeah, I think there's a chance that, right. So we don't really know how specific the signature is, correct? Yeah. So I think there's that risk as well. And I think that, again, in terms of the kind of last point, I think it would be helpful to use more food-specific biomarkers linked to each of the dietary components. Yeah.
Okay, here's the question.
Thank you, impressive work. I'm thinking more of like other cohorts, sometimes RCTs that would not have FFQs or dietary data, where perhaps such an approach can provide a quasi signature of the diet. Have you tried maybe looking into cutting down the 67 metabolites into a smaller number, and if would that really dampen the association, or like maybe 10 or 20, so that it would be feasible for, especially we're hearing how hard it is to reproduce the same signals in different labs?
Yeah, so we haven't actually tried to cut the number down. I think that this was probably, the number that was needed, at least, because again, I think that we needed a panel that was just representative and overall kind of pharmacodynamic change in common metabolites in response to the diet. So I think we needed, a lot of representative signals as opposed to fewer. But I see your point, though. Yeah.
Thank you.
Okay. Well.
Okay.
All right.
Let's thank our speaker. All right, I'm going to tell you a little bit about some of the work on dietary biomarkers that we've done in my lab in collaboration with some great colleagues at the Fred Hutch. Let's see. And this is a long-running study called the Nutrition and Physical Activity Assessment Study, or NPAS, and has several goals. One is to try to identify biomarkers of macro and micronutrients. And then a major goal of the project is to use these putative biomarkers to correct food frequency questionnaires, which Lorraine and others have talked about, and we know that they have a number of issues. And then ultimately to try to take these biomarkers and apply them to large populations and improve disease risk prediction. And the IMPASS study has been, as I mentioned, going on for actually quite a few cycles. The PIs on the project have included Ross Prentice, who was the original PI, Joanna Lampe, and most recently Marion Newhouser is still ongoing. And there are really three major components of the IMPASS. One was a feeding study that included 153 subjects, women between the ages of about 50 and 70, which is a limitation, but the samples basically come from the Women's Health Initiative, and so that is what we have access to. One unusual feature of the feeding study was that the women were all provided, basically they ate their habitual diets, and those were then measured in terms of how much they ate, what they didn't eat, and so forth. And so while the diets were provided, they tried to match up as much as possible to what the women individually were eating, as opposed to many dietary studies where it's more of a food challenge, let's say. And that has both good and bad aspects. One is that there's really isn't much of an adjustment for someone to continue to eat what they normally eat. On the other hand, we didn't have as much control over the range of diet. So we weren't feeding these women Big Macs versus Mediterranean diets in order to really sort of stretch the dietary score, let's say, which would have given us access to, probably better correlations with the biomarkers, because we would have had more of a range. And also, in general, these women were relatively healthy, and so they ate pretty well, and so that was also a bit of a limitation. The second part of the study was 450 subjects. There's part of an extension study where we collected both metabolomic data, but also food frequency questionnaires and a lot of other data, and we used those women to develop corrections or calibrations of the food frequency questionnaires. And then we also had a nested case control study of about 1,500 subjects chosen from the Women's Health Initiative sample sets to try to look at disease risk in colorectal cancer and breast cancer, in that case without the use of food frequency questionnaires. We did metabolomics on both blood and urine, and we had both, depending on the study here of the three. Sometimes we had access to both spot and 24-hour urine. Sometimes we just had spot urine. The feeding design was relatively involved with quite a few different time periods and sample collections of various types, questionnaires, and so forth. We also did doubly labeled water measurements as well as urinary nitrogen, and those turned out to be pretty important. And then this is a summary of the types of metabolomics data that we collected. We did targeted lipidomics, targeted LCMS, aqueous metabolites for serum, and for urine we did a combination of NMR and GCMS. And we had good reproducibility and sizable number of metabolites. However, the GCMS was not as reproducible. And if we were going to redo the study, we would try to ameliorate that. But that's what we had in 2015. In order to develop biomarker panels, then we looked at the cross-validated R-squareds for using lasso methods to come up with metabolite profiles. And our criteria, which is a little bit arbitrary but has served us quite well, is to require a biomarker panel to have an R-squared value of at least 0.36, so that's a correlation of 0.6. And we were able to identify biomarker panels for protein density, so as divided by the total energy, and for carbohydrate density for using metabolites only that met our criteria. And then when we added the doubly labeled water, which is the gold standard for energy intake, then these went up quite a bit. And so then we were able to get both total intake of protein and carbohydrate, obviously energy as well from the doubly labeled water, and then the protein and carbohydrate densities as well. In general, we found that urine was a better sample than blood, but both contributed to these profiles. Spot urine was less reliable, but that's often what you have, and we were still able to make use of that. And so then one of the major goals of the project is to sort of calibrate these food frequency questionnaires, which we know tend to be somewhat nonlinear, where people underreport, especially if someone has a high BMI, they tend to underreport, and they tend to overreport if they have a low BMI. There are also issues of the diet quality that also come in as well. And so the idea of the metabolite biomarkers is to basically correct these self-reported food frequency questionnaire data and then come up with calibrated measures. And so that's basically what we did, and Lorraine nicely summarized the approach, which was basically to develop biomarkers in the ANPAS feeding study, then calibrate food frequency questionnaire data in the extension study, and then apply it to our nested case control subjects. And these are kind of a summary of some of the most interesting results from the study. We compared the food frequency data for hazard ratios of breast cancer, colon cancer, heart failure, and type 2 diabetes, as well as several subcategories of other types of cancers and ischemic stroke and so forth. But the most important results, I would say, here are in some of the cases we were able to get reasonably good hazard ratios just using the food frequency questionnaires, but the hazard ratios tended to be quite a bit larger when we calibrated the food frequency data. And what that indicates is that there is, and it's still not shown in all studies because a lot of studies depend primarily on food frequency questionnaire data, but often, for instance, reductions in carbohydrate levels can reduce breast cancer or heart failure risk and colon cancer risk as well. And these hazard ratios are quite a bit enhanced when you calibrate the food frequency questionnaires, indicating that the risk of these cancers, heart disease, and type 2 diabetes are lower when you have a higher carbohydrate diet, and those effects may be larger than what is currently being measured with food frequency questionnaire data only. I think that overriding this, of course, is the energy intake, and so you have to realize that changing the mix of carbohydrates versus protein and fat and so forth is important, but actually total energy is probably the most important risk factor in these, in terms of diet. We also recently completed a study of looking at red meat consumption and metabolite biomarkers that were, again, obtained using the same data set. And one of the things that, kind of a take-home message here, when we tried to develop these biomarker panels a priori, just looking at the over 1,000 metabolites that we measured across the several hundred samples, in the food study, 153 samples, it was actually difficult to develop that model. So what we did instead was to go to the literature and look at the food biomarkers that have already been established, and found those that we were already measuring in our panel. And then when we did that approach, then we were pretty successful in developing a biomarker panel that had over the requisite 0.36 R-squared value. And then as Lorraine mentioned earlier, we found that we could measure risk, increased risk of, for instance, non-fatal myocardial infarction, coronary death, and total coronary heart disease, which were enhanced in general over just using the food frequency questionnaire data itself. So in this case, we were able to, using already established dietary biomarkers, develop an approach that indicate that the red meat hazard ratios are enhanced for these diseases and may be useful going forward in terms of measuring these in larger populations. So then to conclude, we've been able to use this approach to develop macronutrient biomarker panels for carbohydrates, protein, and by subtraction fat as well, which is currently, we weren't able to do that just using the metabolites alone, but through the subtraction that met our criteria of 0.36. We like to use both serum and urine. We've shown that they're very useful to correct and calibrate food frequency questionnaires and show enhanced hazard ratios in a variety of different diseases. I think that going forward, identifying more of these metabolite biomarkers of specific foods will be very, very useful, and they can be incorporated, I think, very successfully into risk profiles that should be useful, and hopefully they can be boiled down to something that's relatively simple to use and can be applied to large studies. There's still a variety of challenges. We're very interested in getting a total energy biomarker panel, but that's been pretty elusive so far. And we are working on trying to expand the biomarker panels that we have to see how well they reproduce in other studies. I think that's possible, but it's also challenging because many of the studies that we have access to were run under different conditions, different populations, different feeding patterns, and so forth. And so that's one of the challenges. And I will conclude with that. So thank you for your attention. Okay, so we now get to the results of this survey. Oh, I'm sorry. Answer. We have one more survey. Sorry.
I'm collecting the answers, and I am studying right now. So, but not everybody has submitted.
I don't think I did, but.
If you would like to submit, now's a good time.
I have to finish my survey. Just give me a second.
Felt like an election center where, the results don't always different. The winner is science. Okay, so I want to refresh. I don't know how to show it there because it's a link that I sent it and I don't know how to share it, but I do. There's a public link that I can share with people who want to send me an email later and share the final result. Okay, so we have still with question 11, we have not everybody did that, so I'm still waiting another minute maybe.
Oh, I'm not sure if we can connect this computer.
So that's an interesting one. Maybe we can connect my computer. That's a good one. It should be impossible. Huh? Can I just plug this in? plug this out here and plug this in here? Yes? Okay. All right. That's a regular USB. Oh, yeah. And how do I share my screen? I want to share my screen. Okay. Oh, that one. And that one goes in. There. There you go. Have the freshest results, if that's possible. Can we? No, no. How do we share that? Otherwise, I would do it by PowerPoint like that before. Still shows this, your screen, your default. Well, then okay. If it's too complicated, I just put it into a PowerPoint. I already done that, so. What do you want to change? Sorry for that. We could also start discussions, regular discussions, Dan, if you want to take it to the microphone and do some Q&A instead. Oh, all right.
Why don't we have the speakers come up to the front and get ready for that?
Now we can see it? Yeah, let's show. All right. So, yeah. Yay. Whew. Oh, technology works. Fantastic. So thanks for taking part in the survey. It will be shared. Have you read papers on dietary biomarkers? Yes, regularly, some people say. With 35%. Occasionally, 37%. No, not really. Not more than 1 or 2, or never 28%. So that's okay. So most people in the room are interested occasionally or regularly in food dietary biomarkers. That was just to get going, right? Then, do we need dietary biomarkers to better interpret human cohort studies on diseases? The overwhelming majority, 75%, yes. That's why we want to do it. We want to do it on disease risk. And then 4% said, I was interested in this, but no. Food frequency biomarkers are just fine. The questionnaires are just fine. 6%, I've never thought about it, but that's why I'm here in the room. That's okay, right? And 14% says, well, you know what? We want to look at extremes. That's the first part, right? Before we go into disease risk, let's go to extremes, whatever that means, right? Could be vegans and I don't know what extremes are. So that's interesting. Should we focus on human plasma or serum, or more on urine and fecal samples? So 8% said urine is the best because it can be sampled at home and includes already many dietary compounds. 17% said plasma serum is best because it can be easily integrated into clinical studies. 4% liked fecal samples, all right. I don't know who these are, 2 of them. And 70% like, yeah, just sample everything. Sample everything you can and then good luck. Which is a good study design, right? I mean, just sample first, ask later. I guess it's a good idea, to my opinion. So now we go to the topic of chromatography. Are reversed phase LC methods sufficient, or do we need Hillick and others? Together we had almost 20%, 19%, saying reverse phase is fine, it's easiest to standardize and sufficient because dietary compounds are mostly lipophilic anyway, all right? But the vast majority said either other methods are needed because dietary compounds are actually pretty hydrophilic often, and B, we need a multitude of methods, unfortunately, to get the most comprehensive view. That's together is 80% of the people here in the room. This one I found interesting too. Have you shared your metabolomic data with open access repositories like GNPS, metabolites, or other methods? 15% say, yes, I share all my data. Good to you. Great. Good for you. Keep on going and take the others with you. 33%, yes, I have shared data when requested, but I had to. 24, no, I don't. 7% find it too difficult. It's really cumbersome. And astonishing, 22% has never thought about it. Okay, now maybe you want to think about it. Just because 22%, 78% at least thought about it. So the other 22, you should now think about it. Okay, so then the compound ID. Are they sufficient to track and target dietary biomarkers now? This doesn't add up to 100 because mark all that you see fit. 6% said it's fine. With current libraries, we get enough identified. 11%, yes, with identified compounds and accurate mass MSMS repositories, it's good enough. 44, nope, not good enough. We usually identify less than 10%. That's kind of miserable. 29%, no. We need much better computational methods. So if you take these two answers together, it's already a lot. 64, 71% of people said, no, we need a lot better methods. And then 40% really say we need chemical standards to confirm. So basically, 40% of the people here in the room think, don't trust the computational guys. That's an interesting, so we should tell them that, right? That yeah, computational is fine, but verify is better. 13% is I can't decide, I don't know enough, I don't feel safe about it, so I'd rather not have an opinion, and that's okay too. All right, then we had the databases that was mainly FoodDB and related resources by David Wishart and the PTFI. 30%, an astonishing 30% knew about PTFI before. That's remarkable, but hey, that's also illustrious folks here. No, I did not know any of those, was 14%, and yes and no, 57%. That's what I expected, right? Because PTFI just gets going. The official launch was 8 weeks ago, something like that. Yeah, a couple of weeks ago, 6 weeks ago. April something. So, yeah, some of you may have known them before the official release, but now it's officially released. Okay, so how can these repositories be useful for you? I will test them when I get back to work. 46%. Yay, we have achieved something in this workshop. We gave you something that might be useful for you. I mean, ta-da. I mean, how good can it be? No, not my field of interest, but, you know, interesting. I won't. 10%. I already use one, or at least one of them regularly, 30. I think these repositories need much more data before they can be really useful, 16%. All right, so it feels like an election. I like voting. Okay, what is the missing content in repositories on dietary biomarkers? Mark, all you agree. Okay, so that's a lot more than 100%, of course. Let's put it this way. Current repositories are sufficient, 0%. That's a clear vote of no confidence. Right? So let's put it this way. You know, when you ask people on repositories to present what they do, they usually say, we are the best, we are, yeah? So you know these people, right? They go up there and they get a full 0% here. Okay, so I can't decide or don't know enough about it, 14%, that's all right. 57% said more studies are needed across sex, age, BMI, different diet habits, ethnicities across continents, and you name it. Yep, that's why we're here. Many more foods need to be included. 500 is clearly good, but 5,000 would be better, I guess. I don't know what that means. More foods, much more MS data. And compound these across all foods. 55%, even after we heard on the PTFI and the FoodDB methods. More details of specificity and sensitivity of biomarkers to mark food groups, 48%. That goes to Lorraine and, I forgot your name. Yeah, sorry. Last drug states group. Sorry for that. So half of the people in the room basically say so, right? Much more specificity. And Clary said that too. And then too. So, all right, do we need to focus on biomarkers that come directly from foods, or can we use any metabolites that change in response to intake of a specific food or diet? Compounds are directly derived from foods are best, 12%. Any measurable profile that is linked to food intake is useful, 56%. It's complicated. That's usually my answer. Right? It's complicated. That would be I don't know. I was surprised by that. Maybe that's what we can discuss, right? You know, any measurable profile. The vast majority here say that's. I didn't think that would be the majority. Okay. And the last but not least, what do you think after all this kind of workshop thingy about the state of the art? Okay, overall. 5%, that's 2. It was 0% while most of the time, right? So GNPS Massive have a paper out there that says we can already do it. And also, you know, like, can you? I mean, and why are we here? So that was interesting. So 2 people here would agree. We go back. While interesting, we do not have enough resolution, 12%. To detail the food use. Using chromatography mass spec is insufficient. We need NMR as well. That goes directly to Dan Raftery's heart, 17%. I didn't even vote for that. You didn't even vote for it? Okay. I didn't vote actually either. And so then an exciting avenue ready to be used in large studies, 22%. Half of the people in the room say, the missing link to understand many diseases, yay, half of them, you know. We may soon identify typical diets of subjects without asking them, so not you know, even after what Lorraine said, not now. Lorraine really said, if I paraphrase you, you know, just by the markers you can't, but it can help, and adjusting the answers. That's what I heard. But 33% feel the future is near to get rid of FFQs, all right? And a whopping 68% said, overall, most food compounds remain unknown so far. That's why dietary biomarkers are difficult. Okay, so that goes to my heart because it's a compound ID. So that is what you guys thought, and now I thought we have time for another 10 minutes of Q&A.
Thank you very much, Oliver. We have time for Q&A now, so if anybody would like to ask some questions. I know Phil has a burning question. And a microphone, great.
Yeah, thanks everyone for your contributions this morning. One topic that hasn't been discussed so much, though, is this issue about ultra-processed foods, so-called simulated food products that are quite prevalent in many countries these days. Do you have any comments on how to assess that, whether it's artificial sweeteners or preservatives or other indicators of toxic exposure to these types of products?
I can maybe start on that. They're a very diverse group of foods, so it's very difficult at the moment to have biomarkers for them. There are a number of projects that I know that are ongoing where the people are starting to try identify potential biomarkers for them. It's a very complicated field, I think, the whole area of ultra-processed foods. Should we even call them that? I don't know. But not there yet, for sure, but an interesting area.
Yeah, and I'd just add, I'm not an expert on this ultra-processed foods at all, but one thing that I was interested to learn was that the ingredients can be substituted quite a bit, the sources of like syrup or whatever, so it makes it, I think, particularly difficult.
Just, we have looked a little bit at this in the PTFI, looking at complex meals versus highly processed things. I wouldn't call them like single foods, like syrups or these kind of things, more like a Big Mac versus a lentil burger, this kind of thing. And I think there was a hope that we would see a lot fewer compounds in the processed foods or the fast foods, but it didn't really significantly turn out to be the case. These are still complex matrices with a lot of molecules in them. It's really more about what's in them that I think is that may unify the classification more than diversity of compounds.
Wait a minute.
Yeah, just a quick question for you, Dan, regarding the example of red meat and when you calibrated and then the hazard ratios were more significant. So can you give us more details on how exactly you used that calibration or how was it done? Like you went to the literature, you said there were a couple of already markers of meat. So did you use them all? Did you use one? How was that process?
So when we originally built the assays that we used, we weren't planning to look at red meat in particular. And so the process was, first we tried just to build a biomarker panel like we did for, say, carbohydrate or protein, where we were reasonably successful, and it didn't work. And so then we went to the literature and found as many biomarkers that we could find, and then looked on the data that we'd already had acquired. Did we have anserine? Did we have carnosine, and so forth? And where we had those metabolites already measured, those were the ones that we put in for the model.
In what statistical method? I'm just interested.
It's lasso, yeah.
Lasso.
So these are correlations.
Thank you.
Yeah.
Hi, I'm curious about the R-square that you mentioned, like Dan and Lorraine mentioned about the R-square of 0.3, 0.4. I'm curious, like, if it's something that we need to be satisfied with, or if there's something with the calibration. For example, if there is any saturation effect instead of, so it's not a linear dose-response relationship between the intake and the metabolite level. Do you have any comment on that? Thank you.
Yeah, I think the first question was about your R-squared cutoff. Was it? Yeah. first part. I can.
Yeah, so I'll tell you a little bit about the thinking about that. We wanted to pick some value that we felt that would be a threshold that was reasonable, with the expectation that we're never going to get a perfect correlation. On the other hand, we don't want to use any value. And so 0.6 for the correlation or 0.36 for the R-squared value seemed to be an intermediate value. And basically, my colleagues at Fred Hutch, Ross Prentice in particular, would like to, through consensus in the community, establish some sort of threshold value. And based on, a number of studies that he's done, 0.36 we felt like that was a reasonable value. Could it be 0.38? Could it be 0.4? Could it be 0.34? We could argue about that, but that's basically how it was decided. So a correlation of 0.6.
Just comment on the calibration curve that you asked. Does it have to be completely linear? So if you have one biomarker and you're trying to get intake from that, you want to have it linear per se. But if you have multi-markers, so you have multiple biomarkers, not every biomarker then in the panel would need to have such a nice, we say calibration curve is what we had with proline betaine. You might have a couple of markers in there that are very specific for the particular food, and then you move to a multivariate model as opposed to a univariate model.
Just you guys. Really interesting talk. So I just have a question about the metabolome likely represents the complement of somebody's dietary intake and the interaction with their lifestyle and their genetics. So what is the possibility that we're actually going to be able to generate these models for food items or food groups when there's probably multiple interacting things that are sort of coming down on one marker?
I can take this. Well, I think that you're right. There are many different things touching upon what goes on in the body, and this is actually why we put up this biological variability criterion as a next step. And we have, all the ADME that goes, microbiota related. So essentially, the best biomarker would go through untouched, right? Then we have a lot of them that, at least in urine, we have different glucuronides and different phase 2 metabolites that are also dependent on different enzymes levels. So this has to be checked in experimental, well-conducted studies where we expose people with the same food, same people to the same food longitudinally. And I know there are some attempts in Sweden at the moment to have a big cohort on that, so we're excited to see that forward. Did I answer?
I have two questions. Thank you for the great speakers. The first question is, what is a better biospecimen that's better correlated with blood or fecal samples? So fecal or blood is a better biospecimen to be correlated with food frequency questionnaires. Which one would you suggest?
I think, unfortunately, I would say that, at least based on my experience, and we've done a fair amount of stool studies, but never in the context of food frequency questionnaires, so I don't really know the answer to that question. Oliver may have some opinion, but we really haven't done enough work there to know.
Well, I also think what our all the partners said who've done these food frequency biomarkers is that you need often very many people. And if you ask many people to reliably provide fecal matter, there's a practical problem. So that's maybe one of the reasons why people haven't done that so much, because it is much harder to do than plasma in the clinic or urine, even by sending urine samples or so. So I think that's part of the answer. It's not easy to do good fecal studies because it's also very heterogeneous. So, while the amount of fecal matter can be quite a bit, it is so heterogeneous that you might get different parts, a bit different answers. And, there's many practical problems with it. That's maybe part of the reasons why it's not done.
I'm not sure that we would actually want to find the best sample that correlates with the food frequency data. It's really what is the best sample that correlates with the intake, right? So, I don't know the answer in terms of fecal samples. We found that urine is better for a large, at least for macronutrients than serum. But both are useful and they're best in combination.
I've had 2 lives as a career, one in lipidomics, one in glycomics, and the answer is totally different for the two. In your question, humans absorb 100% of the lipids you feed them. They don't get to feces. Whereas in glycomics, if you're looking at fibers, the glycosidic linkages and the sugars that are there from fiber are by definition from the diet. So I think it's not an answer where one is better than the other. It probably depends very much on what you're interested in and whether it has a path to show up in that matrix.
Another question is, I don't know food frequency question enough, so I don't know what's the extent of exposure it catches. Say the red meat biomarker, is it going to be the same for those who just ate beef stew versus those who just ate a high temperature barbecue steak? Would it account for the polycyclic aromatic hydrocarbon carcinogens? Because if we're looking at red meat marker for the health outcome, would it be able to consider those other exposure that's in the food but not necessarily food?
So we don't have I don't have a necessary answer for red meat, but we do see, for example, when we look at potatoes, so we have markers of deeply fried potatoes and mashed potatoes, so you have markers of heated processes, and where biomarkers can actually play a better role than other dietary assessment tools is that you can include these processing biomarkers within the combined markers, so we can then differentiate between what type of or how your food was processed. This is very well in line with the ultra-processed foods, if you want, because then we will need some biomarkers of refined foods as well, which what ultra-processed foods are mainly. So I think they're also refined markers, and trying to get biomarkers of these different types of food processing would be really important in the future.
I was just going to say, I think your question's a good one, though, because the preparation of the food really can have a big impact on the relationship to health outcomes, right? So I think that's an important part of the biomarkers to understand, to have biomarkers of, say, overcooking or high heat, these types of things, yeah.
Thank you.
Yeah, those are really good questions. I was just thinking, and since we were in Japan, if there's been a feeding study of Kobe beef massaged rice or grain-fed well-treated cows, what the outcomes would be in disease outcomes in that case compared to other meat products. I actually had a question about many of the feeding studies is also done on certain populations that are generally healthy, perhaps. And I would question that fat absorption is actually impactful for a subset of people with whether it's IBS or IBD, cystic fibrosis. There's many conditions where you might not get a very different profile in terms of bioavailability than you would in a controlled study of a healthy or otherwise population. Any questions on how to adjust for certain demographics?
It's very difficult to cover everybody in your feeding study when you're doing a controlled feeding study. So you are going to be limited in that instance of finding the biomarkers, at least by who you have in your study. But that doesn't mean you can't go into a cross-sectional study or an independent study and try to see, well, are there now covariates that I should be including that do it? So that's the way we sometimes overcome it. So we do our small feeding study acknowledging that we're limited, then go into a larger population, measure those biomarkers and see, are there covariates that are influencing those biomarkers then that we should be including in our equations?
As we are wrapping up, I have one more comment to that question. We have published last year a method how to sample samples from human cohorts in the upper intestinal tract. We are currently doing a clinical trial with small intestinal bacterial overgrowth patients compared to controls. And so I can give you more on that in about 3 months.
And I think with that, I think we're going to wrap up. We're about 5 minutes over time. So thank all the speakers. And thank you for attending and answering the surveys.